everyone and welcome back to our YouTube channel Playmicrobiology. This is our second lecture on identification of bacteria. In first lecture we have already discussed about the protocol of working in laboratory, like how we work in a laboratory from receiving a sample to culturing. Okay. In this lecture we will discuss about the biochemical test that we apply after culturing. Okay, so let's revise in a short, in brief, what we have discussed in our earlier video. So first we receive a specimen. For example, we receive a urine sample. Then what we will do, we will take a proper culture media for urine, that is clad. Now we will make a well with the help of sample and then streak it. Incubate it for 24 hours at 37 degrees Celsius, and then after 24 hours what we will found? We will found small growth of bacteria, that is called colony, okay, on this entire Petri plate. Right. Now what we will do? Now we will look at this Petri plate, identify a proper colony. We will select proper colonies on this Petri plate that we have to further analyze. Okay, for example, I selected this colony. Now on what basis you will select a colony? You cannot select any random colony from this Petri plate. There are some parameters that help you to identify and pick up the appropriate colony for further diagnosis. Okay, so these parameters are called morphology of colony. It contains 9 parameters almost. So there are there is one mnemonic, means it is SAC HD. Okay, I remember it with the help of second, like second word. Instead of N we will use here H, and that is the only difference. I think, I know there is a big difference in spelling, but it's a mnemonic, just to learn, okay, just to remember the morphology. So there are 3S, 2E, 2C, 1H, 1D. Okay, means there are 3S, 2E. S means shape, another S means size, another one is surface. Okay, first of all we will discuss about size. So size means after looking at colony you will decide the growth of colony, whether the growth of colony is pinhead or pinpoint. Okay, the difference between pinhead and pinpoint is, take your pencil and just mark a small point on your notebook. Then this type of colony, just this small, very small point, this type of colony is known as pinpoint. Okay, pinhead is a little bit larger than pinpoint. Okay, this is the basic difference between pinhead and pinpoint. Next we have shape. Shape means after looking at colonies you will find out whether what type of shape does it have, whether they are in circular motion or whether your colonies are irregular. Okay, next we have surface. Surface means your colony have which type of surface, like they may be shiny or they may be dull. Next we have E. E means elevation, and second E means edges. Okay, edges means whether your colony have regular edges or they are crenated, entire, lobate. Okay, these are called edges. These are type of edges. Okay, next we have elevation. Elevation means whether your colonies are elevated, means whether they are raised, whether they are flat. Okay, what type of colonies do you have in your Petri plate? Next we have C. 2C. 2C means first one is color and another one is consistency. Okay, color. Now as the name shows, color. Color means there would be something production of color. So when the bacteria grow in a Petri plate, they might form colored colonies or it may be possible that the whole color diffuse in your agar. Okay, so first one is they might produce colored colonies. Okay, you might have colored colonies, means your colonies might look like, for example, in McConkey agar, E. coli produces pink color colonies. Okay, E. coli produces, because they are lactose fermented, they produces pink color colonies, but there are many bacteria which do not form any color. Next we have pigment. So there are many bacteria. One more example you can write, Cornybacterium diphtheriae. Okay, Cornybacterium diphtheriae produces black colored colonies. Can you imagine black color, black colored colonies on potassium tellurite agar, tellurite agar. Okay, so this is like whether your colony produce color or not. It is also one of the parameter to select the proper colony for further diagnosis. Next we have pigment. It comes under color. Okay, pigment means there are some bacteria that produces certain type of pigments. For example, have you ever seen mutant agar? It is almost a light yellowish in color, right? But what happens if we have sample, for example, we have sample of pus. Okay, there are 50 to 60% of chances that pus may contain Pseudomonas aeruginosa, Pseudomonas bacteria. Okay, simply Pseudomonas bacteria. Now what does this bacteria do? This bacteria produces different kind of pigments. Okay, from that one pigment is what it does, it color your whole nutrient agar from light yellow to green. Like after growth you will find out that the nutrient agar which was yellow in color, it would convert into greenish. Okay, this is the meaning of pigment. This type of pigments are called diffused pigments. Okay, diffused means the pigment that will diffuse, that will spread into entire culture plate. Okay, so this is the example of this. Next we have consistency. Consistency means like whether your colonies are mucoid, whether they are friable or butyrus. Okay, this is consistency. Mucoid is something like oily. Okay, so this is also one of the parameter of selecting appropriate colony. Next we have H. H means hemolysis, right? Hemolysis, as the name indicates, hemo means hemoglobin. Hemoglobin is present under RBC. And next thing is lysis. Lysis means breakdown. So what will happen here? If bacteria produces an enzyme that is called hemolysin. Hemolysin. Okay, not lysis. It is lysin. I-N in the end. So the bacteria that produces hemolysin would break down the RBCs. What they will do? They will break down the RBCs. Now from where these RBCs come? We uses blood agar to grow our bacteria. Okay, this blood agar contain blood. Blood contains RBCs. From here RBCs came. Okay, there are certain type of bacteria that contain hemolysin, that contain enzyme hemolysin, that breaks down the RBCs present in blood agar. Okay, and what will happen there? Like for example, here is your colony and it would be present on blood agar, right? Then what would happen? The bacteria would lyse the RBC. So here you would see a clear petri plate, like a white surface from the behind. Okay, so this would happen here when it will lyse the RBC. Now there are three type of hemolysis. First is alpha hemolysis, second is beta hemolysis, and third one is gamma hemolysis. So alpha hemolysis means partial discoloration. Partial discoloration. partial discoloration. Partial discoloration means there would be lysis, definitely there would be lysis, but it would not be complete. There would be partial, means incomplete lysis of RBCs, means incomplete lysis. And one of the major factors through which you can identify whether this hemolysis is partial or complete, that is, if there is alpha hemolysis, then there would be greenish discoloration also, means there you will find out green color also, okay, greenish discoloration. So if you see, like you have a blood agar, you have a colonies on it, now you will see the greenish discoloration and you're able to see incomplete discoloration, okay, there is incomplete lysis and there is also greenish discoloration, then you will say that the bacteria produces alpha hemolysis, okay. Next we have example, it is seen in pneumococcus, okay, pneumococcus bacteria. Always underline the bacteria, never forget to underline it. Next we have beta hemolysis. So in beta hemolysis there is complete discoloration, there is complete breakdown of RBCs near the colony, okay, there is a complete clearing of RBC near the colony, okay, NTC short form, near the colony, okay. So in this there would be no production of color. You can easily identify whether it is beta or whether it is alpha, okay. In beta you will see a large halo around the colony, would be large clearing of blood agar near the colony, okay. Next we have, okay, example, Staph aureus, and some students write it S aureus. This is not correct. Never write Staph aureus as S aureus, okay. It is written as like this, Staph aureus. If you want to write Staphylococcus in short, then you can write Staph, but don't write only S. It doesn't mean anything, okay. And in the similar way, some in the similar way, some students write E. coli. I have also written it here somewhere, okay. This is also incorrect. If you are writing E. coli, then write it ESCH, okay, coli, Escherichia coli. This is the correct one. I think it is not visible, okay, this one, okay. So last one we have alpha hemolysis. Alpha hemolysis means there is no breakdown of RBCs. If there is no breakdown of RBC, then it is alpha hemolysis, okay. If there is incomplete partial breakdown of RBCs, then it is called alpha hemolysis. If there is complete breakdown of RBCs, then it is called beta hemolysis. An example is Staph aureus. If you don't have any kind of hemolysis, there is no breakdown of RBCs, then it is called gamma hemolysis. This is all about hemolysis. Last one we have D. D means, do you know what does D stand for? D stands for density, okay. D stands for density. Whether your colonies are opaque, whether they are transparent, whether they are translucent, this comes under density. Transparent means, you all know I think what is transparent, right? Translucent means semi-transparent, from which you can see you can see both sides but it is not clear. That is translucent, means semi-transparent, okay. Next one we have opaque. Opaque means from which you can't see on another side. The colonies that are transparent, the colonies that are translucent, the colonies that are opaque, all are correct for further identification. Like you can pick from these colonies, like you can pick any colony from these three for further diagnosis. Okay, so these are the parameters of colony identification. These are the parameters of, like we can say, this is the morphology of colonies.